Students, today I am going to teach you the biochemical mechanism of cell injury. There are 10 headings in this, 10 mechanisms, and I will deal with each of them one by one. The first one is ATP depletion, second one is the increased cytosolic calcium concentration, third is mitochondrial damage, fourth is ROS accumulation, fifth is nuclear damage, sixth membrane damage, seventh is ischemia, eighth is ischemia perfusion, reperfusion, ninth is toxins, and 10th is radiation. Now you can see most of the mechanisms are interrelated, means to say that one mechanism is giving rise to the other, which can be seen in this slightly complicated figure. One mechanism is giving rise to the another mechanism, about which I shall summarize in the last. Now I will deal with first one by one different mechanisms. The first mechanism is the ATP depletion. Now the causes of ATP depletion you can see are ischemia, mitochondrial damage, and increased cytosolic calcium ion concentration. Ischemia produces hypoxia, which causes ATP depletion. Mitochondrial damage, once mitochondria is damaged, there will be no formation of ATP, again ATP depletion, and by increase of the cytosolic concentration of calcium ion, there is activation of different enzymes including ATPase that breaks down the ATP, and that also gives rise to the ATP depletion. These are different causes of ATP depletion. Now, what are the effects of ATP depletion in causing cell injury? ATP depletion causes inhibition of the sodium-potassium pump. This is combined pump. This is UABane sensitive pump, which is throwing out sodium from the cell and pumping in the potassium from the extracellular space to the cell. So this is its normal function. When there is ATP depletion, which is this active pump becomes paralyzed, and by that there is increased influx of sodium, and sodium is accompanied by water, which causes the cellular injury, and there is increased efflux of potassium. The second mechanism is the inhibition of active calcium pump. Active calcium pump also needs ATP for its function, and this pump throws out the calcium from the cell to extracellular space to maintain a certain level of calcium inside the cell, which we shall deal later. And by inhibition of this pump, again there is increased influx of calcium ion inside the cell that increases the cytosolic calcium ion concentration. Now next one effect is the increased misfolding of proteins, which also occurs by the ATP depletion, and there is detachment of ribosomes from rough endoplasmic reticulum and from polysomes, and that hampers the protein synthesis. Lastly, there is increased concentration of AMP, adenosine monophosphate, inside the cell because ATP depleted, adenosine triphosphate is depleted. Now increased concentration of AMP inside the cell activates two kinds of the enzymes. One is the phosphofructokinase, another is phosphorylase. The increased activity of phosphofructokinase increases the anaerobic respiration, especially in the liver and less in the brain, and increased activity of phosphorylase causes increased glycogenolysis and decreased intracellular concentration of glycogen. When there is increased anaerobic respiration inside the cell, there is increased accumulation of lactic acid inside the cell that decreases the pH of the cell. Now the effect of this one, decreased pH of the cell, are twofold. One is that it causes the increased clumping of nuclear chromatin that causes the nuclear damage. On the other hand, there is the activation of lysosomal enzymes, which causes necrosis. Now on this side you can see increased misfolding of the proteins causes accumulation of misfolded proteins inside the cell, inside the endoplasmic reticulum, and that causes increased ER stress that stimulates the process of apoptosis. So these are all effects of the ATP depletion and causes I have already dealt with. Now I will go to the second mechanism, that is increased cytosolic calcium ion concentration. Now the intracellular concentration of the calcium ion is very low, very low in comparison to the extracellular concentration. It is less than 0.1 micromole, and if the concentration of the calcium ion increases inside the cell, from this one different effects are there which we are going to discuss. First we shall see how it is caused. What are the causes of this mechanism? First cause is again ATP depletion, as we have seen earlier. This causes inhibition of the calcium pump, and that causes increased influx of calcium. That is one source of increased cytosolic concentration of calcium ion. Second one is the damage of mitochondria and a smooth endoplasmic reticulum. These two organs are the organs which sequester the intracellular calcium so that the cytosolic concentration of calcium may be maintained at the lower level, as said earlier, that is almost less than 0.1 micromole. So this is the mechanism how the cytosolic concentration of the calcium ion is maintained low. When it is increased, as by these two mechanisms, what happens? The effects are these. First effect is activation of all the cellular enzymes which normally not remain active, but once the calcium concentration increases, all these enzymes become activated. These enzymes are phospholipases, proteases, endonucleases, and ATPase. So when phospholipase is activated, there is breakdown of phospholipid. When there is protease activation, there is breakdown of the protein. Since membrane is constituted by the phospholipid and protein, so these two give rise to the membrane damage. On the other hand, you can see when there is endonuclease activation, there is DNA breakdown, and that causes nuclear damage. When there is ATPase activation, there is again ATP depletion. So these things are related, and these are the effects. Besides that, second thing is the increased cytosolic concentration of calcium ion increases the mitochondrial permeability transition for pore formation. This causes leakage of the mitochondrial proteins like cytochrome C into the cytosol, and this activates the caspase enzyme present inside the cell, and this gives rise to the apoptosis. So these are different causes and effects of the increased cytosolic calcium ion concentration. Now we move over to next category of the mechanism, and that is third category, mitochondrial damage or mitochondrial dysfunction. How it is caused? It is caused again by the decreased oxygen supply. It is produced by the toxins. It is produced by the exposure to radiation, and also there is decreased survival signals which gives rise to another pathway. So once there is dysfunction and damage of the mitochondria, what happens? What is the effect? The effect is ATP depletion again, increased cytosolic calcium ion concentration, and increased ROS production. These three together causes necrosis. On the other hand, when there is decreased survival signal, means increased pro-apoptotic protein and decreased anti-apoptotic protein. These two give rise to the increased formation of mitochondrial permeability transition pore and increased leakage of the mitochondrial proteins into cytosol. This mechanism, as you have seen earlier, gives rise to the apoptosis. So this is different varieties, different causes and effects of the mitochondrial damage. Now next mechanism is the accumulation of reactive oxygen species, that is the ROS. The reactive oxygen species is especially iron, which is regularly formed inside the cell, normally in the mitochondria, with each electron transfer in electron transfer in the electron transport system. Some of the electron transfer are ineffective, which gives rise to the ions. But these ions or these electrons are effectively removed by the scavenging system of the body. So which consists of the superoxide dismutase, catalase, and glutathione peroxidase, and other antioxidants like vitamin E. These function to scavenge all these normally produced ions and electrons. When there is deficiency of this scavenging system, or when there is increased production of these ions, in that condition there is accumulation of the reactive oxygen species inside the cell. Now you can see what are the mechanisms which stimulate the formation of this ROS. First one is again hypoxia, second is toxin, third is radiation exposure, and fourth is inadequate ROS scavenging system. And what are the effects of this mitochondrial increased ROS? So all these mechanisms, the first, second, and third, causes the mitochondrial damage, and by that there is increased production of ROS. Increased production of ROS, less scavenging system gives rise to the accumulation of ROS inside the cell. Now you can see the net effect of this one is the lipid peroxidation. Lipids present in the membranes, they undergo peroxidation, and the proteins present in the membrane also break down. These two together again give rise to the membrane damage. And on the other hand, the ROS is also causes protein modification, that causes the protein misfolding, and there is DNA damage which gives rise to the nuclear damage. So these are different effects of accumulation of reactive oxygen species inside the cell. Now we will move to the another mechanism, and that is nuclear damage. The nuclear damage, you can see what are the causes. Many we have dealt with earlier, that will be once again repeated. ATP depletion decreases the pH of the cell, which causes clumping of nuclear chromatin and causes DNA damage too. Too severe to be repaired. When DNA damage is too severe that it cannot be repaired, then it gives rise to the apoptosis. Next one is the toxins and radiations. These two increases the formation of reactive oxygen species, and these two may give rise to the direct damage of the chromosomes and DNA, or it may produce and this is known as the genotoxic stress, when chromosome and DNA is directly damaged. Another is ROS production may produce the oxidative stress, that is less presence of more reduced substances in the cell and less oxidative substance, that is oxidative stress, and there is protein misfolding. So these three mechanisms, genotoxic stress, oxidative stress, and protein misfolding, which gives rise to the ER stress, endoplasmic reticulum stress, all these three are responsible for apoptosis. Apoptosis is also stimulated by, as you have seen, DNA damage when it is too severe to be repaired. These are different types of the effects of the nuclear damage. Now next one is the ischemia. As you know, the ischemia is caused by obstruction of arterial blood supply to organs, which decreases oxygen and also decreases the supply of the substrate to the cell. And the second one is the obstruction of veins of the organs. So obstruction of the veins of the organ, what do? They cause inadequate removal of metabolic wastage, while decreased supply of oxygen and substrate increases the anaerobic respiration in the cell, and again ATP depletion. Next variety is the ischemia reperfusion. So Ischemia perfusion is a condition when ischemia, that is partial blockage of the arterial blood supply to the cell, when it is reperfused. The ischemic tissue, when it is reperfused, it gives rise to a different type of the injury of the cell that will be dealt with here. The causes of this one is, for example, ischemic damage of cells increases the influx of the calcium into the cell due to the membrane injury, which increases the cytosolic calcium ion concentration and activates the enzymes as you have seen earlier, and this causes the necrosis. Second is the reoxygenation of the reperfused tissue also increases the electron transfer very rapidly in the mitochondria, which increases the production of ROS, and these two causes, again as you have seen, membrane damage and nuclear damage. Next point here to be noted is somehow the ischemic tissue has deposition of IgM antibody. It is by some mechanism yet unknown, and when there is reperfusion of that ischemic tissue, the complement binds to those IgM antibody and it causes the inflammation causes the cell injury. So reperfusion after the ischemia is another mechanism of cellular injury. The eighth mechanism is the membrane damage, and you have seen from the very beginning how many ways are there of membrane damage. The causes are the ATP depletion, the ROS accumulation, increased cytosolic calcium ion concentration. ATP depletion causes defective membrane permeability of the cell and cellular swelling by the inhibition of the sodium-potassium pump. The ROS accumulation causes injury to the phospholipids and protein, and increased cytosolic concentration causes activation of the enzymes, by which also the activation of phospholipase and protease disrupts the phospholipids and proteins. All these together do the plasma membrane damage and cytoplasmic organelle membrane damage. So this membrane, how it is damaged and what are its causes and how it is damaged, you have seen. Next variety, the ninth one, is the toxin. Toxins can be many type. Few examples are cyanide. For example, cyanide causes injury by the uncoupling of oxidation and phosphorylation. The mercuric compounds which combine with the sulfhydryl group of the protein present in the membrane and do damage. They especially damage those cells, those organs, cells of the organs of the body which concentrate mercurial compound more, and those organs are kidney and GIT. Next example is the production of toxic substances by metabolism inside the liver. What happens? The non-toxic substances like carbon tetrachloride, when they are metabolized inside the liver by the liver enzymes, they are converted into more toxic carbon trichloride ion. Similarly, the less toxic paracetamol or acetaminophen is converted into more toxic substances, both of which are the hepatotoxic. So these are all different mechanisms of cellular injury, and at last I would like to emphasize that I would emphasize that you can see in this figure different mechanisms are interrelated. Like ATP depletion is caused by ischemia, it is caused by toxin, and ATP depletion itself gives rise to the increased cytosolic calcium ion concentration. It can give rise to the nuclear damage, it can give rise to the membrane damage, mitochondrial damage, may give rise to membrane damage, may give rise to the ATP depletion. The ROS accumulation gives rise to the mitochondrial damage, and ischemia reperfusion also may cause the membrane damage. The radiation may give rise to the mitochondrial damage and ROS accumulation. Ischemia also causes the increased cytosolic calcium ion concentration. So all these are interrelated. So Your conception must be clear about all these mechanisms and how they are interrelated to each other, one giving rise to the other. Okay, this much is for today. Thank you. If you have any question, you can ask me in the WhatsApp group or in the YouTube comment section also. I will reply to that. Okay, thank you.